My name is Tobias Walther. I am heading the section of molecular signal transduction at NI Ph.D. And it is my privilege to welcome today's speaker, Dr. Tobias Walther. Dr. Walther is an HHMI investigator and newly appointed program chair of the cell biology department at Sloan Kettering Institute in New York. He is also a professor in biochemistry at Cornell Graduate School of Medicine. Some of you might remember and even be surprised that Dr. Walther is not at Harvard, because he just recently moved from Harvard, where he was a professor at the Chan School of Public Health and an associate member of the Broad Institute. These positions he held until last year. So this has been a busy year for him because he moved to Sloan Kettering, where he is running a lab together with Dr. Robert Farese Jr. They have been running their labs together for a longer period of time. They investigate cellular lipid metabolism, in particular the mechanism of neutral fat production and storage. More broadly, his lab is interested in how cells regulate the supply of fat and the abundance of lipids, how the cells store and mobilize lipids upon need, and how these processes help cellular physiology and membrane biology and dynamics. Of course, it is important that all of these functions are related to human diseases, so they are very involved in pursuing those. They use multiple approaches, some biophysical, some imaging, biochemistry, proteomics, and lipidomics, to discover global insights into the regulation of lipid metabolism. Dr. Walther is a native of Germany. He received his undergraduate degree in molecular genetics from Ludwig Maximilian University in Munich, Germany. And he did his Ph.D. studies in biochemistry at the European Molecular Biology Lab in Heidelberg. He did a postdoctoral study in the UCSF between 2002 and 2006, and then returned to Germany to a Max Planck Institute as a group leader for a short period. He then moved back to the US in 2010 and became an associate professor in cell biology at Yale. From there, in 2014, he joined Harvard and the Broad Institute. I think this is just enough to list all of these movements, so I'm really impressed by the amount of movements you did, but it seemed to be all going upward. So aside from recognition from the HHMI Institute and several NIH grants and awards, I only have to introduce his talk, which the title of this is The Phase of Fat, Mechanism of and Physiology of Lipid Storage. Please welcome Dr. Tobias Walther.
Welcome, thank you so much. I would like to thank you, Tomas, for the introduction and all of you for showing up for this seminar today. It's a great pleasure to be here, and I think I have hopefully the next couple of minutes to hopefully impress you with anything else but my ability to move. So before I get started, I wanted to briefly mention something that Tomas already introduced, which is all the work that I'm going to present today is the result of a scientific partnership between myself and Bob Farese. We work together for nearly 15, almost 20 years now, and work as a scientific partnership. And if you're interested, we recently try to reflect on our experiences here a little bit on how it is to run a lab together. Now, what I wanted to introduce to you is sort of really wonder for a second how much, life really requires in terms of energy. It's just, if you take a regular human being, we generate about our body weight in ATP, obviously it's turnover, but roughly our body weight in ATP every day to allow us to do all kinds of wonderful things, move around, digest, think, whatnot. So, and of course that is balanced by energy intake. You got to fuel the system in order to allow all that to happen. Now, in most systems, can't quite figure out which microphone I'm using here, sorry. In most systems, of course, this balance that needs to happen doesn't perfectly occur over time, but there are fluctuations both in the need to expand metabolic energy and in the ability to take up energy from the environment. And because of those fluctuations, there is a need to buffer temporarily the ability to expand energy, and we do that predominantly by temporarily storing metabolic energy. And by and large, this occurs in the form of lipids. This here is drawn roughly to scale for a healthy human and shows roughly how much metabolic energy we temporarily store in the form of lipids versus carbohydrates. And presumably, this has evolved as a near universal form of metabolic energy storage because the molecules here, the lipids, are stored in the form of triacylglycerol, a molecule that's characterized by a glycerol backbone that has 3 triacyl chains esterified to its backbone. And that makes that molecule extremely hydrophobic, so it doesn't mix with water. As a consequence, this is the densest form you can store carbohydrates in, right? Because now you don't have the water come in. And presumably, as I mentioned, this is the reason why this has evolved to become more or less universal form of energy storage. But it creates a problem. So because biochemistry happens predominantly in aqueous space, but when you put triglyceride in an aqueous space, phase separation occurs. The oil doesn't mix with water, that's the whole point, and there's phase separated from each other. And you can reconstitute this biochemically in a cocktail glass on my porch. And you can see that oil, of course, doesn't mix with water. So here's where all the energy is stored in a cell, if you will. And the water is really where all the life happens. And at the interface, a lot of biology needs to happen to allow the position of the energy into the oil phase, withdraw things. And some of you may remember that in old days, in order to extract DNA, we actually used such interfaces to denature all the proteins and get rid of them to extract the DNA in the water-soluble phase. So this here creates a unique challenge. In cells, the oil is not stored as a sort of massive layer of fat, but instead in what we now know as organelles that we now know as lipid droplets. Those are very unusual organelles compared to, let's say, mitochondria or the endoplasmic reticulum, in as much as they contain a core of neutral lipids that forms an organic phase, which is bounded by a monolayer, not a bilayer membrane, of phospholipids into which specific proteins are embedded. And really the discovery of by Dean Landos and others that there are specific proteins to this surface really started to put this organelle onto the map as something that's not just an inert blob of fat, but an organelle in cells with all the usual dynamic behaviors. You can see that lipid droplets interact in cells with other organelles, such as mitochondria or the endoplasmic reticulum as well. And if you take a single lipid droplet, right, it can store about, on the back of the envelope, if it's about 300 to 500 nanometers, is what we often observe, it can store the equivalent of some 23 billion ATP equivalents. So that's really where a lot of the metabolic energy is temporarily stored. And as I mentioned, in cells, you have multiple of these lipid droplets most often, and their presence converts cells to emulsions. Really, the cytosol is a continuous phase. The lipid droplets form a dispersed phase here, and surfactants, phospholipids predominantly, are there to basically delineate those two surfaces and allow all the traffic in and out and all the biology that I'm going to talk about. Now, in multicellular organisms, we have further separated and specialized the function of energy storage from other tissues, and predominantly fat is stored in adipose tissue. And what you can see here in this slide is like this here is a unilocular single lipid droplet that occupies most of the adipocyte, and then all the other organelles are sort of squeezed to the side in this cell type into this sort of dark gray surrounding. But what's important to notice is that lipid droplets are not restricted to adipocytes. They occur in pretty much every other cell type people have looked at. This includes, for instance, after a meal, the intestine, where in blue here you see lipid droplets accumulate after fatty acids come in from the meal. It also includes the liver, where fatty acids are trafficked to from either a meal or the adipose tissue and temporarily are stored in lipid droplets. The heart predominantly runs on fatty acids as a metabolic fuel and temporarily can store lipid droplets. And for instance, of course, milk has a lot of fat. Those are lipid droplets that get shed from the epithelial cell in a still very poorly understood mechanism that is fascinating. So lipid droplets are really present throughout physiology, and not only in physiology, but also in pathophysiology. So, for instance, many of you know, of course, that we are facing an unparalleled epidemic of obesity in this country and elsewhere, where really massive accumulation of triglycerides interfere with the physiology of humans. What's a little less well known is that there is also sort of a different conditions known as lipodystrophy, where a failure to establish or maintain adipose tissue leads to severe disease. And in both cases, one can think of, for different reasons, that adipose tissue gets overwhelmed with fatty acids. And although this person here looks phenotypically somewhat healthy, actually those two conditions share very similar hallmarks in terms of their metabolic and endocrinological disease. Now, on the biochemical level, triglycerides are synthesized by essentially two enzymes. And they are known, they were initially discovered by Eugene Kennedy and co-workers in the 1950s, and they were purified and then named after their activities. So they're known as DGAT1 and DGAT2 for diacylglycerol acyltransferase 1 and 2. And those two enzymes basically take a diacylglycerol, which is a polar molecule in a hub in metabolism that can still form phospholipids and other things, and make it an apolar triacylglycerol. And after Bob had discovered particularly DGAT1, it turned out that this is a fantastic drug target if you're a mouse. So if you're a mouse, it turns out if you delete or inhibit DGAT1, the mice live 30% longer, they're resistant to diet-induced obesity, and they have essentially all positive characteristics of metabolism. So this has inspired a lot of pharmaceutical companies to develop inhibitors, and I'll say a little bit about that in a second. But it turns out that when you think about triglyceride synthesis, there's two parts to this equation that are worth to consider. One is that these enzymes make triglycerides and put them in lipid droplets, but they also remove things such as fatty acids and diacylglycerol. And those are two sides of the coin here. So when you, for instance, generate a mouse that does not have both enzymes specifically in adipose tissue, this mouse is surprisingly healthy. It doesn't have any particular problem. You can see that it has adipose tissue, but the adipose tissue doesn't have any fat, so it doesn't swim up, in this case in an EM fixative. It's just a simple way to visualize the effect. And these mice are fine under most conditions as long as they have food. But if you don't have food and you put them, for instance, in the cold, which is a major metabolic stressor in mice, they cannot maintain their body temperature. Right? So this will argue, as you probably know, and what I argued for is that triglycerides really function as an energy source. But what also happens is that in many cases, such as obesity, this excess lipids overwhelm the machinery to form lipid droplets and therefore flow into other metabolites that, when accumulating in the ER, can trigger ER stress that either leads to cell death or adaptation. And it turns out in humans, that's what happens in the intestine, because those mice that I mentioned express DGAT1 and DGAT2 in their intestine, and we only express DGAT1. And when we discovered patients that have mutations in this enzyme, they have a severe congenital diarrheal syndrome that results in a protein-losing enteropathy and a fat-dependent diarrheal syndrome. And essentially what we think is going on here is that when the organism is confronted with a lot of fatty acids from the meal, the enterocyte cannot temporarily buffer
that influx in lipid droplets, and as a result, this triggers enterocyte dysfunction, ER stress. And consistent with this, we now can figure out how to deal with this with bypassing the intestine for those patients. More broadly, this has led to a concept proposed by us and others that lipid droplets really have a protective function in many contexts, such as the liver, macrophages, skeletal muscle, and heart, where they often protect the tissue from the accumulation of metabolic disease. And we think that this is also why DGAT1 inhibitors failed as a drug, because they, in the intestine, essentially kill that protective function. Also consistent with this, we now know from work of many laboratories, including ours, that the processes involved in lipid storage are intimately linked to inherited metabolic diseases, because when you have mutations in enzymes involved in the synthesis of triglyceride or the formation of lipid droplet organelles, you get a number of severe diseases. Now, with that, you could ask, is all hope lost? And it turns out there's also inhibitors for this other enzyme. And there's a lesson here, because when we initially found that enzyme and made a deletion, this was lethal in mice. Mice die soon after birth. But it turns out that now this has become a front runner for the treatment of fatty liver disease, again, because of a species difference. And we find that if you just restrict the effect to mice, to liver, that protects the animals from fatty liver disease, one of the major side effects of obesity, because now the fatty acids are not only in the adipose tissue anymore, but they spill over to the liver. This protects from that and has led to moving those drugs into clinical trials with markedly reduced liver fat. This is initially done by Pfizer, but also by Ionis. And it turns out what I told you shouldn't make sense, right? Because you still have this protective effect. So why does this not become a problem when you inhibit DGAT2? Well, it turns out you not only block triglyceride synthesis, but you also have a feedback regulation that shuts off the synthesis of more fatty acids and thereby prevents essentially the accumulation of any lipid toxic intermediates. All of what I told you in a relatively short progression is just meant to give you an introduction that lipid droplets can have sort of two major functions in cells. They can be used to store metabolic energy and to detoxify potentially harmful intermediates of metabolism. What I really want to focus today on is sort of three topics in the basic biology of these organelles. I want to talk about how are the triglycerides really actually made in the endoplasmic reticulum? What are the enzymatic mechanisms? How do you then package them into the lipid droplet? And how are the proteins that mediate the exchange with the rest of the cell actually getting there? And finally, a little bit about how do you actually extract and mobilize things from a lipid droplet again. So in the first part, how are cell synthesized triglycerides? I already told you that there's two different enzymes, but I haven't told you that those two really only share a name. Otherwise, those are evolutionarily unrelated, DGAT1 and DGAT2. DGAT1 is a polytopic membrane protein, and DGAT2 has this sort of hairpin domain and otherwise is cytosolic. We recently solved the structure of DGAT1 by cryo-electron microscopy, and what this revealed is a butter-shaped dimer here. And if you focus on one side of it, what we found is that the catalytic center for this enzyme is deeply buried within the plane of the ER membrane, inaccessible from both the cytosolic side through a tunnel and also through a lateral gate within the membrane. And based on many experiments, we now believe that this catalytic center is accessed from the side of the cytoplasm by a fatty acyl-CoA, looking like this little carrot here, that accesses the catalytic site. And from the lateral gate, a diacylglycerol moiety accesses the catalytic center, esterification occurs, and a triacylglycerol molecule is formed that can then be released into the plane of the membrane. Intriguingly, and just sort of as a side note, this enzyme is a member of a family called MBOATs, from membrane-bound O-acyltransferases, which is a large and fascinating group of enzymes that can acylate lipids, but also are involved, for instance, in the formation of important signaling molecules such as hedgehog or wingless. We now have structures for a number of them, but I will skip this. We have much less information on DGAT2, but somewhat interestingly, It was one of the major proteins highlighted in the initial AlphaFold prediction. It was in the main figures to show that this is how we think this enzyme is looking like based on the prediction. And intriguingly, it has an active site actually not in the membrane, but on the cytosolic side of the membrane. So now that we understand that particularly this enzyme here releases the triglyceride in the membrane, the next question becomes, how then do you form a lipid droplet after the triglyceride is released into the membrane? For many years, the general thinking was, well, you just release it into the membrane, and we know from work of Hamilton and others that there is a maximum solubility of triglyceride in a phospholipid bilayer around 3%. So up to 3% of triglyceride, they just solubilize in the membrane. And they're, if you think about it, very similar to the side chains of a phospholipid. But above 3%, phase separation occurs and an oil phase starts emerging. And so for many years, it was thought that that's just a passive process that occurs. But we and others have found that actually cells don't leave this process up to chance, but instead have evolved dedicated protein machinery that organize the formation of lipid droplets out of the ER. Specifically, a key protein for this is a protein called sapin, named after one of the discoverers of a mutation in the 50s that leads to this lipodystrophy. So this protein is called either sapin or BSCL2 for Bardet-Biedl SAPE congenital lipodystrophy. And the mutations in this protein essentially lead to a complete lack of adipose tissue. And this is thought to occur because during differentiation of adipose tissues, massive synthesis of triglycerides occurs. And when the machinery that I will show you involved in triglyceride packaging into lipid droplets is deficient, you get ER stress, toxicity, and presumably that's why those cells don't survive. It was then, like, some almost 50 years later, this protein was, the gene encoding this protein was cloned. And it was shown by a number of labs that the deficiency in this perturbs lipid droplets, for instance, in yeast, plants, worms, and so forth, many, many different labs, including our own, where we found initially in a screen in flies and later in mammalian cells that the gene encoding this is a representation of a genome-wide screen looking for phenotypes associated with the depletion of each gene, gives one of the strongest phenotypes on lipid droplets. So that phenotype can be summarized in short. This is in a fly cell here, that in normal cells, there is a few pre-existing lipid droplets. The lipid droplets here are shown in this green color surrounded by a lipid droplet marker that we call life drop. You can see that there's some pre-existing droplets, and then when you add fatty acids to the cell, the equivalent of the McDonald's diet, we're really driving this thing. You can see that there's a few lipid droplets forming in an organized way at some sites in the lipid droplet, in the ER. When you do not have sapin, however, there's again a few pre-existing droplets, but when you induce the formation of new ones, you can see that there's this whole snowstorm of these very small blisters of lipids occurring everywhere in the ER. So that phenotype is consistently observed across different species. So how does this protein do that? So we tagged it at its endogenous locus with a fluorophore and tracked how this looks like. And here you can see an ER signal in red and sapin tagged in green. And what immediately becomes obvious is that the protein forms foci or complexes in the ER membrane. And again, you can see this easy in a Drosophila cell, but it's the same is true in a mammalian cell, in this case a human cell. That, of course, immediately brought us to, like, okay, what are these complexes? How do they work? What do they have to do? And why do you get that phenotype? So in order to understand that, we solved the structure of such a complex. And you can see this here. This is by cryo-electron microscopy. And what immediately becomes obvious that this forms a very large oligomeric ring structure in the endoplasmic reticulum membrane that is characterized by a luminal domain, right, sitting in the lumen of the ER, and many transmembrane segments. So to give you a little better view of this, what you can see is that there's sort of a cage structure that is again open to the plane of the membrane and otherwise sort of in this conformation relatively closed. And what we think happens here is that this provides a space in the center of this, not unlike, for instance, GROEL or during protein folding, where triglyceride molecules can diffuse in through those lateral portions and find a space that excludes phospholipids and thereby helps catalyzing phase transition, or in other words, the likelihood of 3 triglyceride molecules to meet rather than interacting with a phospholipid. We have tested this idea in molecular dynamics simulations where we find, for instance, here these are together with Si-Young Kim and Greg Vogt at the University of Chicago, where we find that only when sapin is present here, red, lipid droplet formation occurs within the center of this very large complex. And then you can see sort of in these molecular dynamic simulations that the complex opens and the lipid droplet forms towards what would be the cytoplasm. And importantly, we can see that only when we have sapin can you start nucleating lipid droplets or the phase transition of triglyceride at concentrations below that solubility level. So at least in these simulations, that appears to be a crucial part of this molecule to help with the nucleation. Now, when you look more careful at that structure, as I mentioned, it has sort of a ring of luminal domains with hydrophobic helices protruding into the membrane. We think that this actually helps exclude phospholipids, and these are very important, as also shown by others, for the nucleation and transmembrane domains. What we did, however, realize is that these hydrophobic helices that push apart the phospholipids are not only conserved in their character, but also in evolution are conserved in their specific sequence. And that is somewhat unusual, right? If you just need to be hydrophobic, you're just hydrophobic. So why is there any specific sequences conserved? So Jaeyeon Chung in the lab postulated that we're maybe missing a cofactor. And she did some experiments that I'm just going to show one of here, where she either had these little hydrophobic helices protruding into the membrane or didn't have them, did a pull-down experiment and found only one protein really that specifically interacted with this form of the complex, but not that one. And that was uncharacterized. It was a membrane protein that we then have renamed from TMEM159 to lipid droplet assembly factor 1. And what Jaeyeon could show is that when we take cells where we tagged sapin at its endogenous locus and this new protein LDAF also tagged at its endogenous locus, and then we follow lipid droplet formation by a third color knock-in and using a marker protein, that lipid droplets really only form at sites that have both sapin and this LDAF protein. And you can, yes, so that's why we renamed those two proteins together, form the lipid droplet assembly complex. And one of the things that was sort of gratifying about this is you might imagine, right, if sapin is so important and sufficient and would be sufficient to mediate this, that it interacts with triglycerides. But when we purified sapin biochemically, we never found any associated triglycerides. Only when you co-purify and reconstitute the complex with LDAF, now you can see that triglyceride associates with this, and we know that it's triglyceride because it is sensitive to the inhibition of the synthesis enzymes and can be raised if we add extra fatty acids. And structurally, what this does is it actually appears to really start sort of transforming the micelle. If you look at sapin alone, it forms a very flat micelle. But when you have LDAF in it, we see that basically there's two things in here. There's an additional density of protein that's very poorly resolved, and we can talk about it, why I think that is, and also triglycerides. So we think that this is sort of the earliest intermediate of lipid droplet formation that we can visualize, where we start having this complex and some triglyceride trapped in the middle. And fascinatingly, as lipid droplets start forming, the complex disassociates, the sapin stays in the ER membrane, and LDAF1 starts to decorate the surface of lipid droplets. So if I put all this together into a model, what I told you about, it'll look roughly like this. So at the level of the ER membrane, you have enzymes such as DGAT that take the fatty acyl-CoA and the diacylglycerol and synthesize a triglyceride. That then is laterally released into the plane of the membrane. and can diffuse into this cage shape of the sapin-LDAF complex. That allows phase transition. And then once you start that, the complex opens and goes towards the cytoplasm. At that point, additional monomers of triglycerides are integrated into this by a process equivalent to Ostwald ripening, right? So it's easier to get into this phase than it is to get out of it. So that's our current understanding of how lipid droplet works. There's a lot of mystery still here. Now, once a lipid droplet forms, for it to interact with the rest of the cell, it is really important to have the specific set of membrane proteins that are on the surface. And a key question is, like, how are proteins targeted to the surface? And we still don't understand that completely. We believe that this is a fundamental question in biology because it's not similar to other things. There is no import machinery known for lipid droplets, such as for the ER and mitochondria. There are no specific lipids that mark this surface, such as, for instance, for an endosome or the plasma membrane. But instead, the structure of this surface is quite different. And I will tell you about this in a second. And what this leads to is that in proteomic studies, we now know there's upwards of 100 proteins that are specific to lipid droplets, depending on the cell type, et cetera. These proteins come in different flavors. Some of them you might expect, synthesis or degradation of triacylglycerol. That makes sense. They should be on lipid droplets. There's others which you can still rationalize, such as phospholipid remodeling enzymes. And then there's a large class of others. This includes transcription factors that we start to understand are involved in the regulation of metabolism. There is enzymes in there involved in glycosylation reactions that many labs have found, and frankly, nobody knows why they're there, what they're doing there, and whether that matters. If you're interested in studying this, we built a knowledge portal together with the Broad Institute that integrates a lot of that data and allows you to sort of dynamically browse all the information that at least we and some other labs have generated on lipid droplets. Now, how do all these proteins get there? As I mentioned, this is an untrivial problem because we don't really know any of the pathways. So what we have learned so far is that generally there appear to be two distinct pathways. In one pathway, you make proteins in the cytosol and then they directly bind the surface. In a second pathway, proteins are initially inserted into ER and then go to the surface of lipid droplets. In that, and we call them cytold and ertold for short, cytosol to lipid droplet or ER to lipid droplet. The cytold pathway includes enzymes, many of the regulatory and structural proteins, and also regulators, transcription factors, and I will tell you about one other protein later. And how do those guys actually get to the surface is non-trivial because they have a sequence here, an amphipatic helix that has the propensity to bind a membrane. But why do they go to this membrane rather than that membrane that is vastly more abundant? There's a lot more ER surface than lipid droplets in cells, even though they have the same lipid composition. And part of the solution to this mystery is that the underlying triglyceride changes the surface properties of the monolayer. And I don't have time to show you all the data, but essentially, if you imagine a bilayer membrane has to be tightly packed. All the phospholipids have to be really next to each other. Whereas in a monolayer, you can push them apart a little bit, and the triglyceride can push apart the acyl chains just a little bit. That is known as packing defects and provides surfaces that can be read out by these proteins. These packing defects are more abundant, more persistent, and larger on the lipid droplet surface, modeled here in a bilayer phospholipid membrane with some triglycerides under it than in other membranes except very highly curved ones. So what we think happens for many proteins, and I don't have time to show you all the data, is that they have sequences that are disordered in the cytosol but have the propensity to bind the surface through very large hydrophobic residues such as tryptophans, which insert into these packing defects. And then secondarily, this leads to the folding of amphipatic helices that have one hydrophobic and one hydrophilic surface, essentially leaving this process irreversible. Also then requiring actually a different process to get these proteins off the surface later on. How about that other pathway? So some proteins we know are inserted initially into the ER, and then they end up on the lipid droplet. This includes metabolic enzymes, such as this GPAD protein or ACLCL3 and 4. These are enzymes involved in the Kennedy pathway of lipid synthesis. It also includes the now best known risk factor for the development of fatty liver disease from from steatohepatitis, HSD17B13. So how do they get there? What's the mechanism? And of course, even we know that proteins don't jump in and out of membranes, so this can't be it. Instead, what we found is that when you analyze lipid droplet formation and you tag, let's say, you visualize the endoplasmic reticulum in green and one of those metabolic enzymes as a cargo in red, you can see that those two start out in the same place, and overlap in their color. But when you make lipid droplets, the two colors separate, and the metabolic enzyme ends up on the lipid droplet, and the ER stays back. So how does this occur? We know that this enzyme, at least, really ends up on the surface of lipid droplets because we can do immunogold labeling with an antibody against the endogenous protein and show that it really decorates the surface of a lipid droplet, but not the endoplasmic reticulum that is close by. So the first thought, of course, is like, well, it's pretty simple, right? What's the big hoopla, right? You start with a protein that's in the ER, you make the lipid droplet out of the ER, the proteins just go on to the droplet when it starts, right? So something like that. During formation, the protein goes on the lipid droplet. Well, and some proteins actually can do this, such as this LifeDrop probe, which is derived from this enzyme. And you can see that actually during lipid droplet formation, they go on. And for this particular probe, we now know that this is due to a change in the conformation of the enzyme that alters its energy state between the bilayer and the monolayer. In short, our current model is that when these things are inserted into the ER membrane, that insertion anchors the protein to both sides of the membrane, and with that places large hydrophobic tryptophans in the middle of the bilayer, which is highly unusual and energetically unfavorable. And when they come to the lipid droplets, they swim up, that the tryptophans become interfacial, and we believe that that is what drives really how they accumulate. I should say this is all a model because to date there is not yet a structure of a lipid droplet protein. But it turns out only some proteins can do this, right? We thought case closed, done. If you follow the endogenous enzyme and not just a little fragment of it, there's a real mystery. You can follow lipid droplet formation for hours here, and nothing happens. And unfortunately, in this projector, you can't see the green diffuse background signal very well. But what you can appreciate is that for, again, 350 minutes, nothing happened. And then within 10 minutes, one of these droplets here became accessible and targets that enzyme to it, right? And then very rapidly, like in a triggered fashion, this lipid droplet acquires this protein. So how does that work? Well, the first question is like, well, if it was in the ER, as I told you, why doesn't it get in here? Well, it's not hard to imagine that if you have this large megadalton complex at the base of lipid droplet formation, that it doesn't let everything through, and particularly larger cargoes such as this full-length protein cannot go there. And this is actually the case because the experiment I just showed you here, simplified, says that, okay, in a wild-type cell, after half an hour, even though droplets are formed, the enzyme is not on. Later in the formation, it is. If I take out sapin, as soon as droplets form, the enzyme hops on. So the sapin complex prevents some proteins to go on to lipid droplets. But there is a pathway because at the end, they end up on lipid droplets. So that's a real mystery for us. And it led us to hypothesize, well, there must be two pathways. One early that allows the access of some proteins to lipid droplets, and one later one that maybe uses some independent bridges. And it turns out, at least in terms of the timing, there's a whole slew of metabolic enzymes that appear to use that. And you might wonder why so complicated, and I'm happy to talk about that. So it turns out that really there are these multiple membrane bridges. You can show this here. This is a FRUP experiment at an intermediate time point. There is signal already on the lipid droplet. And at the beginning of the experiment, we bleach that. And what you can see is that the signal recovers and that it appears to recover through multiple connections between the lipid droplet surface and the endoplasmic reticulum. Those connections can also be visualized by cryotomography. You can see here ER studded with ribosomes that is directly contiguous with the lipid droplet surface. And here's just this is just colored in for better imagination. Well, that of course then triggers is like, how would you generate such membrane bridges between the ER and the lipid droplet that are independent of that initial formation site? And this we banged our head against for many years, quite frankly, and had a number of ideas, and eventually decided to brute force it. So Jion Chung in the lab decided to do a visual screen, knocking out one gene at a time, every gene in the genome, and asking which ones specifically abolish the recruitment of lipid droplet proteins to this, but not other cargo, either to lipid droplets or other secretory pathways. And what he found was actually a specific class of tethering components, membrane fusion machinery, and recycling of that membrane fusion that is specifically required for this pathway. We can see that those components not only are required, but they're at the right time, because we can do a somewhat complicated experiment here, but just to show without explaining it in too much detail, I'm happy to answer any questions about it, but we set up a system where you can basically temporarily segregate the trafficking from formation. And what we found is that as soon as we basically trigger, these are minutes, in a cell fusion experiment, the potential for the protein to go onto a lipid droplet, right before that, the protein will end up on this lipid droplet. We see that the machinery that we discovered, and this is just one example, will show up right at what will become a membrane fusion site between those two. So our best model right now is that there is an independent machinery that involves ER exit sites and membrane tethers that allows the formation of secondary contacts, or not actually just contacts, membrane bridges between the ER and lipid droplets. And if you think about it, of course, this may make some sense because this allows you to uncouple protein trafficking from lipid droplet formation. There are many cell types that don't actually make a lot of lipid droplets, but they still have to change their proteome, and maybe this is a pathway required for that. However, there's definitely more open questions than answers here. So, we don't understand really how this barrier works. I can tell you that size doesn't matter. It's not the single distinguisher here. We don't know how this pathway here is triggered at all. We don't know how persistent these bridges are, whether they're one-way or two-way streets, and how they participate in physiology. And so we're still busy for a while. But with this, I want to switch gears for a second here, right? So now I told you how you can make a lipid droplet and you can get the proteins on there that matter. All of this, of course, presumably only matters in the context where you also have mechanisms to mobilize fatty acids from the lipid droplet, right? You want to store, put things in the bank, but you need to withdraw them at some later time point. Now, generally, there are two distinct pathways how this can be achieved in all the cells that people have looked at. The first one is that enzymes, such as ATGL, that showed up already on some of my slides, can target the surface of lipid droplets, and they're essentially hydrolases that cleave those ester bonds, and particularly in this case, the first one, and hydrolyze the triglyceride and release fatty acids. It turns out there's like 3 distinct lipases that take the first, the second, and the third acyl chain. These enzymes were discovered, ATGL, particularly by Rudy Zechner, and who has done some beautiful work showing how this works, particularly in adipocytes. It is essentially not understood in any other cell type. There's a second pathway that you can imagine, which works a little different, where a lipid droplet or part of a lipid droplet is internalized into an autophagosome structure. The autophagosome is fused with the lysosome, and then in the lysosome, acid lipases hydrolyze the triglycerides there, okay? And this has been known as lipophagy. This pathway of lipophagy was initially observed, more than 10 years ago. And it was particularly reported that if you knock out autophagy, let's say in liver, you accumulate a lot of lipid droplets. But it has been really, and then there's hundreds of papers after that, right? But it's been really difficult to ask, okay, how much of this happens? How does it really work? How is it regulated? And to me, at least most importantly, how does this contribute to physiology? Because the only tools that we had were blunt, like we knock out autophagy. But nothing has been known specifically. How do you connect the lipid droplet to the autophagosome? What selects a lipid droplet? And to me, that was an interesting question, and luckily also to Bob and Jeon Chung. And while that is an interesting question, the discovery we made was really serendipitous. And it started with Jeon asking, somewhat randomly, for a lipid droplet protein, where is it localized? And this lipid droplet protein is called Spartan. And what was really peculiar was that when we localized this with a knock-in endogenous locus, we found that it localizes to lipid droplets. They are lipid droplets because they stain with neutral lipid dyes. But it doesn't localize to the same lipid droplets as a genuine lipid droplet surface marker, PLIN3. That was bizarre to us. And particularly then we asked, okay, what's different about these lipid droplets than the rest of the lipid droplets? And what Jian started to do is systematically co-localize what might be different. And what she found pretty quickly was A, the Spartan marked lipid droplets always localized next to a lysosome. You can see this here. And they tend to be, and they're almost always marked with a LC3 protein, and a core part of the autophagy machinery. And I will tell you a little bit about Spartan now. So what is this protein? So it is a protein that has a couple of suggested domains. So there is something in the M terminus that's called a microtubule interacting domain, which most likely does not interact with microtubules. There is a ubiquitin binding region that has nothing to do with the ubiquitin binding region that some of you may know from the N rule. And there's a senescence domain that really has nothing to do with senescence, but is found in a genetic screen in plants with mutations in it, somehow associated to a complex phenotype. But what we postulated is that maybe this protein serves, or hypothesized, as a protein that bridges the lipid droplet and the lysosome, or the autophagosome, just based on that localization. And I don't have time to show you all the data, but what we found is that the senescence domain really is composed of a series of repeated amphipatic helices. There's a whole set of them in here. And what we found is that these proteins are in cells sufficient to target a protein to lipid droplets. And this is consistent with work from others, actually, that was published before. In addition, this domain here in the middle really looks like a pH domain, and it can interact in an uncanonical motif as a Lyr domain with LC3. And we've shown directly, I don't have time to show you all of this, that this proteins bind directly in vitro. And so based on this, we think that this protein Spartan might be functioning as a receptor that bridges the lipid droplet surface to the autophagic machinery. Now, and again, the paper on this will be published shortly, so you can have a look at that. But this, of course, then brings up the question, well, does it participate in lipophagy or not, right? Protein-protein interactions are nice, but what does it really do? To do this, we developed an assay where we fused a fluorophore, that's called chema, to an integral protein of the lipid droplet surface. And this is a peculiar fluorophore in as much as it has a different excitation maximum in neutral and acidic pHs. And so you can distinguish this protein as long as it's in the cytosol. It will be has a green maximum, and if it goes to an acidic environment, it shifts to a red maximum. And that is the transition you would expect from something being delivered to a lysosome, of course. So it goes from green to red here. So what we found is, like, if you take cells, load them with fatty acids, and then withdraw the fatty acids to force them to mobilize their stores, much of the lipid droplets appear to deliver to an acidic compartment. And that is blunted when you do not have Spartan. So this is a CRISPR knockout of that enzyme. We know that consistent with this, cells that do not have Spartan accumulate triglyceride, because now they presumably can't mobilize it as well. And this accumulation is not due to a difference in the synthesis of triacylglycerol, but degradation. And the degradation here is roughly on the same scale that you observe with, the effect is on the same scale that what you observe with the mutation of an autophagy machinery, and that's not on the slide. If you mutate both together, there's no additional phenotype. So with all of this, the current working model that we have is that Spartan decorates the surface of lipid droplets and interacts directly with a phagophore machinery, such as LC3, and delivers this protein delivers the lipid droplet to lysosomes for degradation. Now with that, now it becomes possible to ask, okay, where might this matter in physiology? And what I haven't told you yet is, like, why does it have such a funky name? Why is this thing called Spartan? So it turns out Spartan is also known as SPG20, and it is found in human mutations that cause a complex spastic paraplegia known as Troyer syndrome. So that is characterized by autosomal recessive paraplegia with progressive muscle weakness, but also small stature, developmental delays, and a whole bunch of neurological defects. The Troyer syndrome mutations appear to be spread all over the protein, quite frankly, but concentrate also to some degree on this middle domain and somewhere in the senescence domain. Consistent with this idea that it somehow has something to do with neurons, it is highly expressed in the brain and particularly in neurons. Frankly, this is the last place where I suspected lipid droplets to matter, right? If you think about the brain, not necessarily the first place. Adipose tissue, the liver, that's where people look for lipid droplets. What about the brain? So we made initially, as a first experiment, we made neurons in vitro from iPS cells, differentiated them either wild type or Spartan knockouts. And what we found is that only when you don't have Spartan, you appear to accumulate quite a large number of lipid droplets in the cell body. But this is cell culture. These eye motor neurons are, maybe a model of neurons, but they're definitely not the brain. So we developed a system to test this in vivo, and specifically what we did with Jung-Kyu Park at Wayne State is develop a system where we would have, where we'd inject either a control or a dominant negative fragment of Spartan that we developed in vitro, and asked, okay, what is the effect? And we expressed one on one side of the mouse brain, the other one on the other side of the mouse brain. You can see that the control expresses much higher than the dominant negative that we inject, and we asked what are the consequences. And what we found is that specifically cells that express this dominant negative Spartan start to accumulate lipid droplets in their cell body. And then when we go in and cut out those regions specifically, we can see that the brain of those accumulates a triacylglycerol and also some DAG, but that the rest of the lipids are not changed. And so with this, we think really that for some reason, autophagy of lipid droplets appears to matter in those neurons and is required for normal neuronal function. Now that's really surprising, and to my, I really don't know why yet. What is tantalizing to us is that when we go back, what I haven't told you yet is that there are mutations in sapin that are also called SPG17. So they're also spastic paraplegia genes. There are different mutations in sapin that don't lead to the lipodystrophy. These are missense mutations mostly. And they lead to a spastic paraplegia. Spartan I told you about. And somewhat concomitantly, we collaborated with Ben Kravat's lab, who had shown and discovered an enzyme, DDHD2, which is a triglyceride hydrolyse that's brain-specific. And that mutation, so mutations in this is also leading to spastic paraplegia. In addition, there is a whole slew of mutations that are known to give you spastic paraplegia and often affect ER function, but also very prominently affect lipid droplets. So we're now really quite interesting to ask, okay, what might be the function of lipid droplets, particularly motor neurons? And I can't answer that question for you today, but I'm happy to talk about it. So what I told you today, some highlights were that I hope I helped you understand a little bit how we currently think triglyceride synthesis occurs in the DGAT enzymes, and particularly DGAT1, where we have most insight. That there is a dedicated machinery in the endoplasmic reticulum that forms a very large megadalton complex that allows the formation of lipid droplets out of the ER, thereby protecting the cell from lipotoxicity of distributed lipid intermediates. That there are distinct pathways to get onto lipid droplets, some of which we start to understand to some degree, and that there is a fascinating new area of lipid droplet biology with autophagy in the brain, where we start to have some of the molecular players that will allow us, hopefully, to analyze the physiology of all these processes. And with this, I want to particularly thank the people who've done the work today. I highlighted work by Jeon Chung and Henning Arlt. Jeon was a fantastic postdoc in the lab. So usually, right, when you're successful as a postdoc, you go somewhere else. Your mentor stays. In our case, she got a job at Harvard. She stayed. We had to leave. It's too bad. No, she's great, and she'll have a fantastic lab. Jeon Sung did a lot of the work on the membrane trafficking. He was a fantastic MD PhD student. Shuibu Sui did a lot of the structural biology on DGATs and the initial work on sapin, and is now faculty in Texas. And Henning Arlt is an outstanding biochemist at the Dana-Farber now, who worked on the sapin structure. And with that, I also want to thank our funding, particularly the NIH. Thank you so much for support over many years, particularly NIGMS. and also, of course, HHMI and the blue field. And with that, I'll take any questions you might have. Thank you.
So absolutely, people here, please use the microphones. And I'll announce first the CME code, if I remember correctly, 44463. And I guess that's it for now. Please. Matthias.
Very nice presentation. So I have a two-part question. So this Spartan protein, is that specific only for lipid droplets to deliver those to the autophagosome, or do you find it on maybe other compartments or other organelles?
Yeah, thanks for that question. I should have said this. This protein was also reported in a number of other contexts. For instance, in the cleavage furrow of mitotic cells. We tag the endogenous locus and we never see it anywhere else other than distributed in the cells or at lipid droplets. And that is consistent with some other work that has reported that localization, also proteomics. So we have no evidence that it acts anywhere else. But
and what is the signal for localizing to the lipid droplets? Is it like by default sitting there and then, you know, turned on or off?
Yeah, you caught me, right? I set this up nicely in the, like, wouldn't it be nice if we could understand the regulation and I didn't tell you how the regulation works? I don't know. It is, you can speculate, there is a monoubiquitination site in the protein that can be extended to a polyubiquitin chain, and that appears to be regulated by the metabolic state. And it correlates with a change in localization. But we do not know how that works, what triggers it. We know that, you know, there are certain stimuli, but the ones that we have in vitro are not physiological, right? You don't go and withdraw fatty acids. And so we don't know is the short answer. But
Thanks.
it appears regulated. Also, I don't know, right? This protein appears to bind a subset of lipid droplets, but the signal you get is all around. So like one protein binds, I get that. There must be something different about these droplets, right? Why do they all go to that droplet and not the one next to it? And I don't know that either.
You mentioned that the glycosyl transferases are in the endoplasmic reticulum, and whereas of course the plasma membrane thickness is bigger, much more than the endoplasmic reticulum. As the sorting of these proteins takes place according to the, I know glycosyl transferase stay there because they have a shorter transmembrane domain. If you extend that, we have transported those to cell surface, the plasma membrane. So the thickness of these plasma membranes, the composition versus the endoplasmic reticulum, is there a difference that they form this thickness and that the proteins are then embedded into here versus the sorted out according to the thickness?
If I understand your question correctly, you're talking about a concept that's also known as hydrophobic mismatch, right? Where the length of a transmembrane domain is matched to the thickness of the membrane.
Yes.
And the thickness of the membrane tends to increase as you go towards the plasma membrane, right?
Yes.
And that's matched. If you think about it, the lipid droplet is the extreme case of a thick membrane, right? You have one palisade of phospholipids, and then there's the ocean under it, essentially. So we do think that that matters for that mechanism, but it is important to remember you can't presumably have transmembrane domains per se, right? The trans part that gets you, that has one piece here, then there's a membrane, and then there's another piece. There's no other lumen down here. So they're all just dipped. So I think that makes it fundamentally somewhat different.
Yeah, I know when you synthesize this protein glycosyl transferase, your N-terminal end is the transmembrane domain. If you extend that transmembrane domain to this protein will go to the plasma membrane. We have shown it in this.
Yeah. I'm aware of that.
Yes.
So I do not know whether that plays a role here. I did mention, because I mention this every once in a while because at some point someone's going to have a clue. If you just look through literally now hundreds of proteomic studies, like lipid droplets were thankful organelles because you grind up your cells and then you float the lipid droplets on top. And then you can do mass spectrometry. And when you just do that, it probably tells you more about your mass spectrometry abilities than what's on lipid droplets. But there's lots of other things you can do. But in hundreds of these studies, one of the things that repeatedly comes up is N-linked glycosylation machinery. And that's also been localized by microscopy. And to this day, I have zero idea why they're there. You can speculate if you think about the structure of a dolicole, which is important as an acceptor, that maybe that's in lipid droplets, but nobody knows. So that's a long, winding answer for like I have no clue.
Let me sneak in one question online
Sure.
before you go. So in your picture of the encapsulated LD in a lysosome, the two surfaces seem to contact.
Huh.
Does the snare, et cetera, machinery play a role? Why is encapsulation, lipophagy, needed?
So we do not know any other machinery at this point that's required for specifically lipophagy. Presumably, and it is peculiar, right, that what we observe is a co-localization directly of the autophagosome with a lysosome. That is unusual, right? Normally, autophagosome forms and then later it fuses. We don't understand why. The encapsulation presumably is needed to allow for the action of acid lipases. So they've been known for a very long time. So there's a protein called LAL, lysosomal acid lipase, that only works in acidic pH, and that's also been, there's knockout mice for that. They're important for mobilizing triglycerides, et cetera. So that's presumably what it does, but they do not know any of the other machinery that's involved in this process yet.
So that was great. The curvature of lipid droplets change a lot from when they're born to when they're very large. Is there any evidence that that plays a role in activating enzymes that sit on the surface of the lipid droplet?
Short answer is no. Actually, there is, if you go to the biochemistry of these processes, there's still a lot of mysteries. We have not worked on this, so I don't really know much about it besides what's published. But if you think about, for instance, so ATGL, this is a lipase that Rudy Techner has done fantastic work on, really. It sits on the surface of lipid droplets. It's perfectly capable of, it should be perfectly capable of doing what it does, but it's inactive. And then there's another protein that is also on the surface of lipid droplets, and that needs to bind that protein to activate the lipase. Like, why? And then they're both there at massive concentrations, right? But they can't interact with each other because it turns out that thing is also binding something else. So all these things right now are still in the phase where we have essentially some very high-level concepts, but really nobody understands. There are no structures for these things. The biochemistry is really not very well understood. And despite that, there are already drugs, for instance, like the ATGL inhibitors are pioneered for sort of cachexia, cancer cachexia, and other things. So, yeah. So I'm just saying that you're on a good track here. This is super fascinating. I have no answer, and I haven't even an experiment or something like that.
Thanks.
That could help.
Great talk. I like the minutia. Looking at your DGAT1 and 2, you know, DGAT1 having this lateral gate, do you think, you know, versus this cytosolic-oriented active site of DGAT2, is it accessing DG from different leaflets? You know, whether it's in the cytoplasmic leaflet or the inner leaflet?
Yeah, I don't have much time to talk about this yet. So I think the general idea in the field is that diacylglycerols can flip-flop very fast because they have essentially no polar head group. What is specific about DGAT1 is that it is always in the ER, and it takes the diacylglycerol from the ER. We've shown, and many others, actually some in Canada, that DGAT2 localizes also to the surface of lipid droplets. We don't know whether it's in the droplet or in a contact site with the droplet, but those droplets grow. So it is very tempting to think that the DGAT1 is sort of detoxifying the ER stuff, makes the initial droplet, but DGAT2 really sort of locally expands the droplet. Consistent with this, Jay Horton at UT Southwestern has some beautiful work that suggests there's channeling between de novo lipogenesis all the way to triglyceride synthesis, but the cell biological basis for this is not yet known.
Yeah, and that kind of leads me into the quick follow-up, which is, you know, you talked about targeting enzymes to droplets, and, you know, this GPAT4 is the basis of the live drop sensor. You know, you talked about the lipidome of the droplet, but, you know, that's making lysolipids. You know, you don't have AgPAT, which is a multi-transmembrane pass on the droplet. So do you think that, you know, lysolipids are playing a role? You know, this idea of fusion, ER exit sites are also allegedly enriched in lysolipids.
Yeah, these are all really good questions, and it's a little embarrassing that I don't have answers for you. I can tell you that if you just do crude experiments and you pull on GPATs and cells, you will find an ECPAT, and you happen to find the ECPAT that's involved in lipodystrophy. If you pull on DGAT, people can find SEDs, which are the things that desaturate and really important for this. So I think there's a fairly high chance. And then, as I mentioned, there are some people who, Jay Horton particularly, beautiful work on the fatty acid synthase and the upstream enzymes coming together on a complex around lipid droplets. How these, so there's a fairly high chance that there is some sort of cellular organization of this pathway. But there's just a chance. People haven't looked. And honestly, also, like, this field hasn't been necessarily a center of attention for cell biology for many years, and then the tools are only now catching up. If you overexpress these things, usually, you overexpress these enzymes, hell breaks loose and everything's everywhere. So it's very hard to study.
Thank you so much.
So try to make it quick. So the surface of the lipid droplets, because of their non-canonical nature, I would imagine that the proteins that bind and the protein structures are also non-canonical. So I'm curious that, for example, a tool like AlphaFold that bases on training sets is probably rather underrepresented in what you would need to then predict structures of these proteins. Can you share your thoughts about that problem?
Yeah, there are no publicly available structures for a full-length lipid droplet protein. There has been some really beautiful work on lipoproteins, right? The other lipid droplets, much older, much better studied, but they're not like really three-dimensional structures, but they're more like, okay, amphipotic helix, how does this work? So, so because of that, we don't find AlphaFold as valuable for this type of protein that we find it for other things. Like, we've worked on some ER proteins, and I go like, wow, this is amazing. And then, we get like a cryo-EM map, and it just looks like, we're really blown away by the potential of this. We have not had that experience with lipid droplet proteins, where often you get like something that looks like a random coil-ish thing with a bunch of helixes left or right, or where it's really hard to make sense of what AlphaFold predicts.
Thank you.
So I don't think it's that useful yet.
So thank you. We're not going to go, but we do have some other great questions, but we'll get them to you elsewhere from our colleagues. And yeah. Yeah. Not yet.
So I want to thank you again.
Thanks so much. Thank you, everybody.